Hello everyone, and welcome to our lecture on viral and prion diseases. Just like with bacterial diseases, we can't cover everything, but I'm trying to cover the highlights of some of the most significant viral infections in the United States and also worldwide in some cases. And also, just like with the bacterial lecture, again, there are no real unit or lesson objectives here. Your goal is kind of to learn as much as you can about each of these viruses in terms of the information that's on the slide and in the lecture. So, viruses are an interesting group of organisms to talk about because unlike bacteria, we can't always fit them into very neat categories like gram-positive rods, gram-negative rods, gram-positive cocci, gram-negative cocci. The best we can kind of do is talk about what we call viral families and then talk a little bit about their structure in terms of whether they are enveloped or non-enveloped, whether they are DNA or RNA. Now, one thing I will tell you is I will never ask questions on whether a virus is an RNA virus or a DNA virus, whether it is enveloped or non-enveloped. That kind of information isn't as significant or important for you to remember as things like what type of disease it causes, what the symptoms are, how it's treated, how it's prevented, whether there's a vaccine for it, etc. I include stuff like this to help you guys organize it because sometimes that makes it a little bit easier to study or to write notes for or to group things together. But you will not have a question on an exam that says something like, is a herpes virus enveloped or non-enveloped? Is it DNA or RNA? There won't be questions like that. There will be questions about what disease a certain virus will cause, what the course of disease is like, what the signs and symptoms are like, etc. But you don't have to remember this type of information. It's just there to help with organizational purposes. So, all of that being said, let's go ahead and start in on a few of the very important herpes viruses. Most of the time, when people hear herpes virus, they think about either oral herpes, which we also call cold sores, or genital herpes, which is oftentimes a sexually transmitted infection. And that is true, but keep in mind that the herpes viruses are a large group of viruses. There are many different types of herpes viruses. And when we're talking about the viruses that specifically cause things like cold sores and genital herpes, those are called Herpes Simplex 1 and Herpes Simplex 2. Generally, Herpes Simplex 1 causes oral infections, and generally, Herpes Simplex 2 causes genital infections. That's not always true. It is possible to get HSV1 in the genital region, and it's possible to get HSV2 in the oral region. It's just most of the time, HSV1 is associated with oral herpes and HSV2 is associated with genital infections. Now, in terms of the disease this causes, it is usually referred to as herpes. And this virus is one that infects the body and then never really goes away in most cases. So what happens with Herpes Simplex 1 and 2, both of them, is the virus gets into the body some way or another, either through doing something like sharing drinks or kissing or some other kind of oral-to-oral contact or some kind of sexual transmission, and it causes its initial infection. Most of the time, that initial infection is kind of like a blister or a sore, either in the oral region or the genital region, and then it goes away. How long it lasts kind of depends on the person, depends on the dose they were first exposed to, maybe a week, sometimes 2 for some people, but most of the time it clears up within about 2 weeks. It can last a little longer, again, depending on what else might be going on with the person, but within a week or so, most of the time, that blister kind of heals up, the sore heals up, it goes away, and the virus just remains latent in nerve cells. From that, it just stays inactive in the body until some kind of environmental factor activates it again. The exact environmental factor that causes this virus to kind of become active again is still not known. There are lots of things that seem to be associated with it. It can be exposure to UV light. It can be certain types of foods, it seems. It can be just the general kind of catch-all of stress. There are lots of different environmental factors that seem to be able to influence this virus to become active again. Once the virus becomes active again, it starts replicating itself through the normal viral replication cycle, kind of migrates to the skin, and causes another case of what we would call herpes, where just that blister, that sore breaks out in the area, and it is kind of an annoyance, it's an inconvenience, but most of the time it's not serious. That being said, there are some situations where herpes simplex 1 or 2 can be serious. One can be if a woman has genital herpes while she is pregnant and she has an outbreak right before delivery. It can be very problematic for a baby to be exposed to that virus during the birthing process. So there are different avenues that can be explored or different methods that can be done, but oftentimes one of the reasons or one of the things that will be done is a woman will have a C-section instead of giving vaginal birth, just to be extra safe and prevent the baby from being exposed to that. There are other situations where herpes can be problematic as well. Anytime someone is immunocompromised with something else, herpes infections can get worse. Occasionally, although not very often, and this can happen in healthy people too, herpes can migrate to the brain and cause swelling in the brain and brain damage. But most of the time, this is a fairly mild infection, mild virus. Many people in the world have it. I think it's estimated that maybe about 70% of adults worldwide at least carry it with them, even if they don't have obvious outbreaks. So it's just a very common virus for humans to have. And like I said, most of the time, it's an inconvenience, it's an annoyance, but generally not too serious. Another important virus in the herpes virus family is Varicella Zoster. This is a virus that causes both chickenpox and shingles. This is another instance of a virus where it will enter our body, cause disease the first time, and then just remain latent, oftentimes for decades. So the first time someone gets infected with this virus, the virus enters the body, causes its initial course of disease, and that is called chickenpox. So the first time someone gets this disease, it's called chickenpox or varicella. After that, the virus just remains dormant in most people for a long time. Now, it's possible that this virus could reactivate at any time, but for most people, chickenpox is something they get as a child, and then the virus stays dormant until maybe their 50s or 60s, when it will become activated again for various reasons. The exact activation factor we don't know. There might be many. And that second course of disease, when the virus becomes activated and starts replicating, it tends to kind of stay isolated in one part of the body, and it causes what we call shingles. So the first time we get exposed to this virus and get sick from it, most of the time we get a rash kind of all over our body. And then what the virus does as this infection is kind of clearing up is basically picks a couple of places to lodge itself in the body. Usually those are in like the spinal nerves or sometimes the cranial nerves in the facial region. And that virus just hangs out, oftentimes for decades, doesn't really do anything, just sits there in a latent form. And then for whatever reason, it reactivates, and it really only becomes active in one region of the body instead of being spread everywhere. It just kind of activates in one specific region and causes a very painful rash where the blisters kind of pile up all on each other, usually in just one region of the body. Now, there are vaccines for both of these. So there is a chickenpox vaccine that will help people who have never had chickenpox prevent themselves from getting this disease. And there is also a shingles vaccine. The shingles vaccine is for people, for the most part, at least at this point in time. This could change. But the shingles vaccine is for people who have had chickenpox and they are getting to the age where it would be very common for this virus to reactivate. What this vaccine does is help kind of boost the antibody levels to this virus that a person has in their body, so that if the virus that, again, is already in their body becomes reactivated, they have enough antibodies to kind of fight that off. And even if they can't prevent a full resurgence of shingles of the virus, they can at least make it so the symptoms are not very severe. And then our last herpes virus that we'll talk about is one called the Epstein-Barr virus. Most of the time when we hear about this virus, we hear about it in the context of what we call mononucleosis, or mono. What happens with this virus is that when someone gets infected with it, typically it gets into their body through the oral route. They share a drink, they kiss, they share chapstick, or some other kind of situation where they exchange oral fluids. The virus starts to kind of replicate all over the body, and it causes certain white blood cells in the body to form these very big nuclei. That's why it's called mononucleosis. So normally the nuclei of these white blood cells don't look quite this big and large. They're usually much smaller. But what ends up happening with this virus is that it causes the white blood cells to go through certain types of changes, and their nuclei become very large. And in that process, there are a variety of symptoms that people might experience. They can have extreme tiredness, sore throat, fever, swollen lymph nodes, headache, rash, all of those types of things, plus more body aches and other things like that. And then for most people, it seems like this virus tends to go away and not cause any further problems. But there are situations where this virus can make someone more prone to developing certain types of cancers, or it may be related to chronic fatigue syndrome. So it's not definite that this virus is always related to chronic fatigue syndrome or is the cause of it, but there is an association between someone having been exposed to this virus and potentially have gotten ill from this virus, and then later developing chronic fatigue syndrome. So again, I don't want to scare anyone or say that if you've had mono, you will definitely develop chronic fatigue syndrome. That is not the case. That's not what I'm saying. It's just there seems to be a relationship between people who have had this virus and been ill with it may be more likely to develop this later in life. Our next set of viruses that we'll talk about are measles, mumps, and rubella. Now, these are not necessarily closely related viruses, but I group them together because they are all vaccinated against in the same vaccine. So the MMR vaccine is the one that's designed to help protect people from measles, mumps, and rubella, and that's why I talk about them all on the same slide. One thing they do have in common is that they are all enveloped RNA, and remember, you don't have to remember that, it's just to help you organize things if you want to. And most of the time, they cause fairly mild illnesses, but especially with measles and rubella, things can get out of hand and cause long-lasting damage or sometimes even death. So with measles, the way this is spread is it is spread through respiratory aerosols. That means when people cough or people sneeze, they will spread this virus to other people. And one thing to understand about measles is that it is one of the most infectious viruses in the world. So it is spread through the air very easily. It doesn't require a very big exposure to actually get sick from measles, and it is quite easy to pass from one person to another. Most of the time, the infections caused by measles are relatively mild. Someone will get a rash spread throughout the body. They will get a fever. They may develop cold-like symptoms or flu-like symptoms as well, because it is ultimately a respiratory virus. And within about 2 to 3 weeks, most of that resolves on its own, and the person can go back to living normally. However, there are plenty of cases where the virus does not really resolve on its own, and the infection leads to something like fatal pneumonia or brain swelling. Another word for that is encephalopathy. And that oftentimes leads to death because the pressure on the brain becomes too great, the organ stops functioning, and the person dies as a result. So most of the time, measles is a fairly mild illness, but not for everyone, and people do die from it. And as we start to see the decline in the number of vaccinations for this disease and for others, we have started to see a rise in the number of cases of measles, and again, a rise in the number of serious complications as well. Our next virus that we are going to talk about here on this slide is the one that causes mumps. Mumps is primarily spread through salivary or respiratory secretions. So most of the time we're talking about close contact, something like, again, sharing drinks or kissing. It can be spread through respiratory secretions, although that's not quite as common as through salivary secretions. In terms of the illness, the course of the illness, signs and symptoms, many times this is a very mild illness. In fact, usually it's a fairly mild illness. It tends to lead to swelling either in the salivary glands, kind of in the front of the throat area, or the parotid glands near the ears in the jaw area. And oftentimes that'll just be one-sided, so kind of one side of someone's face will swell up really big. And within about 2 to 3 weeks, it typically goes away, the swelling is reduced or the inflammation is reduced, and everything goes back to normal. It can happen on both sides of the face as well, so it's not like it's always on just one side, but oftentimes we just see kind of one-sided swelling near the facial area. And then for males, this virus can also infect the testicles, so it can cause complications with testicle glands as well. And those can oftentimes swell up very large as well and cause a lot of discomfort and a lot of pain. It was thought for a long time that if a boy had mumps as a child, he would not be able to father children later, but it's important to understand that that is usually actually not true, and that males who have had mumps even as children are oftentimes, if not usually, able to go on and have children. So it is not a reliable method of birth control, no matter what anyone says. And then our last virus that we'll talk about on this slide is the one that causes what we call rubella, or German measles. This one is another one that's spread primarily through respiratory secretions, either through oral exchange or through the air when someone coughs or sneezes. And most of the time, this is a very mild illness in anyone who has already been born. So most of the time, as long as you have been born into this world, even if you're just a few hours old or a day old, if you come in contact with this virus and get an infection, almost all of the time you're going to be okay. You will get sick, you will get a rash, you will be uncomfortable, but most of the time, anyone who has already been born will survive this pretty easily. However, if someone who is pregnant is exposed to this virus and they have either not had it before or they have not been vaccinated for this virus, and they don't have antibodies to protect them and the developing fetus, this is another one that can cross the placenta and can start to cause damage in the developing fetus. So this type of infection in a fetus can lead to everything from relatively mild symptoms. Sometimes you see some facial deformities, sometimes you will see stunted growth later on, to much more severe ones where we're talking about things like cognitive delays, brain damage, and even death. So this is a potential cause of things like stillbirth or miscarriage. And again, as we see a decline in the vaccination rate, especially with the MMR vaccine, we have started to see more of a resurgence of rubella or German measles and the associated complications. Our next set of viruses are those that cause viral hepatitis. Now, there's a couple of things to keep in mind before we get into the details of this slide. First, hepatitis is kind of a general term similar to something like pneumonia. or meningitis, and lots of things can cause hepatitis. So when we're talking about pneumonia, we're talking about certain types of lung infections. When we're talking about meningitis, we're talking about swelling of the meninges, those coverings that surround the brain and the spinal cord. And in both of those cases, lots of things can cause pneumonia, or lots of things can cause meningitis. And the same is technically true for hepatitis. So there are ways to get hepatitis that have nothing to do with a virus. However, since this is microbiology, we are focusing on the ways we get hepatitis from viruses. Another thing to keep in mind is that none of these viruses are closely related. So it's not like we're talking about viruses within the same family like we were in the herpes virus slide. In this case, none of them are very closely related. They differ in terms of structure. For example, in terms of whether they're naked, whether they're enveloped, whether they are DNA, whether they're RNA, etc. So there is some variation within this group of viruses. However, we tend to talk about them together because they all do result ultimately in the same thing, which is hepatitis, and basically that's inflammation of the liver. So to start us off, we will talk about hepatitis A. Hepatitis A is typically the least severe infection when we're talking about virally caused hepatitis. Most of the time, if someone gets hepatitis A, this is a self-limiting illness, meaning they will get exposed to the virus will multiply in their body, specifically in their liver, cause some problems, typically for a couple of weeks, and then it will go away on its own. This virus is transmitted through the fecal-oral route, and most of the time people get it when they've been exposed to contaminated food. So this is a situation where someone who is carrying this virus does not wash their hands necessarily well enough after using the bathroom. The virus manages to make it from their hands into food that's being prepared. Somebody else ingests the food, and they get exposed to the virus that way. After that, the virus leaves the gastrointestinal tract. It moves to the liver. And then while it's in the liver, it will start to replicate. It will cause some cell damage. Typically, symptoms are things like abdominal pain. There may be a little bit of jaundice. It's not quite as common with hepatitis A, but it can happen. Maybe kind of flu-like symptoms, a low-grade fever, and basically someone just doesn't feel very well, typically for maybe 2 weeks or so, and then most of the time it goes away on its own. It can become a more severe infection that requires treatment, things like antiviral medications or additional supportive care just to help make sure the person gets through the illness without a lot of damage being done to their body. And the good news about this one is that there is a vaccine for it that most people get sometime during childhood or adolescence or adulthood as well. Just depends on kind of whether they got it as a kid or not. Hepatitis B is the next one that we will talk about. Hepatitis B can either be a relatively short-term illness or can turn into a long-term kind of chronic situation. This one is obtained primarily through bodily fluids. It can be through sexual contact. It can be through things like sharing needles for IV drug use. It can also even be from things like sharing razors or toothbrushes in certain situations. Hepatitis B is another very, very infectious organism, so it doesn't take a lot of exposure to become ill from this virus, and this virus is actually pretty good at withstanding time outside the human body. And so, like I was saying, even things like toothbrushes or razors could potentially transmit hepatitis B from one person to another. For the course of infection, typically what happens is it gets into the bloodstream, gets into maybe the lymph, either way, circulates throughout the body, and then it replicates in the liver and starts to cause damage as a result. In terms of symptoms, some fairly common symptoms are things like loss of appetite, fatigue, fever, abdominal pain, sometimes nausea and vomiting. Sometimes people will experience jaundice, although that's not quite as common, but it can happen. And for many people, this will go away on its own and they won't have any long-lasting damage. But for some people, it will become chronic or at least a long-term infection that does require treatment to make sure that it doesn't cause extensive damage to the liver. There is a vaccine for hepatitis B. It is usually a 3-injection series that people get very early on in childhood. And interestingly, this is sometimes one of the vaccines that is pointed to as an example of how we over-vaccinate kids, because in their mind, hepatitis B is only transmitted through sexual contact and kids are not having sex. So to them, it's an example of how we over-vaccinate. But remember what I was just saying, hepatitis B is not only transmitted sexually. There are many different ways that it can be transmitted. It is incredibly infectious, and so even small doses from potential daily activities could be enough to expose someone to hepatitis B. And that is one of the reasons that it is given to children at a young age, is to prevent that from happening. Now, the last one we'll talk about is hepatitis C. There are actually a couple of other viruses that do cause hepatitis. They're not very common. There's one called hepatitis D, but again, that happens in very, very low numbers. It is a very similar course of infection to the other hepatitises. You may start hearing more about that one in the next decade or so as it becomes more prominent, but for right now, A, B, and C are the most common causes of virally caused hepatitis. So hepatitis C, like I said, this is the one we will end with. Out of these 3, this is the one that is most likely to cause long-term infections, potentially lead to cancer, potentially lead to permanent damage in the body. This one is primarily transmitted through bloodborne exposure, so blood-to-blood contact. It can occasionally be transmitted by other bodily fluids, but the vast majority of cases seem to come from blood-to-blood contact. So it's very common for this to be transmitted through needles when people are sharing needles. It could be, sometimes they've had cases transmitted through people sharing straws. If they're using a straw to snort drugs, basically, and it ends up cutting the inside of the nose, sometimes that will lead to transmission as well. So hepatitis C is not only transmitted through drug use, but it does typically require blood-to-blood contact in some way or another. So a blood transfusion, if the donor blood wasn't screened properly, could also be something that transmits hepatitis C. In terms of the course of infection, this is, again, another one, gets into the blood, makes it to the liver, starts to replicate and cause damage to the liver. And for most people, this is not one that's going to be self-limiting. This is one that's going to turn into a chronic infection that can cause long-term damage to the body. The good news is there is actually a treatment now that will cure hepatitis C. When I first started teaching here 10 years ago, this was a disease that was not curable. It was one that we could manage with certain types of antiviral medications, but it was not one that we could ever fully get rid of in the body. However, enough research was done, the medication was developed, and now people with hepatitis C are able to get a treatment, as long as they have access to that treatment, that will actually cure the infection so that they don't have to live with it for the rest of their life. The unfortunate thing about this one, well, there are a couple of unfortunate things about this one, but one of the unfortunate things about this one is that we don't have a vaccine for it. The surface of the virus and the antigens that it has are pretty variable, and so it's been very difficult to come up with a vaccine for it because the strain of hepatitis C that one person gets may not match the exact strain of hepatitis C that another person gets. So because of the antigenic variation on the surface of the virus, it has been very difficult to develop a vaccine. However, I'm kind of hoping that with the advent of the mRNA vaccine, we may have a better chance of developing a vaccine for this one as well. Our next set of viruses are the papilloma viruses. These viruses are primarily associated with what we call warts, which are small growths or kind of outcroppings, oftentimes, on the body. There are over 100 strains of papilloma viruses that infect humans, and there are all kinds of papilloma viruses that infect various animals as well. So typically you're not going to have a situation where a papilloma virus that infects a rabbit, for example, also infects a human. It's just that there are basically papilloma viruses that seem to infect all kinds of different, especially mammalian species out there. So in humans, there are over 100 different types of papilloma viruses out there. They all are capable, at least, of causing various types of small growths on the body. About 65 of them are really common in terms of causing what we would consider just standard normal warts. 35 of them cause warts in the genital region. And of that 35, there are 9 specific strains that increase the chance of someone developing a reproductive cancer. So of all of the papilloma viruses, they're all kind of numbered. And strains number 16 and number 18 cause about 70% of reproductive cancers. Over 90% of cervical and anal cancers have some kind of viral involvement. And these viruses can also be involved in causing other types of genital or oral cancers as well. So cancer of the penis, cancer of the vagina, cancer of the throat, cancer in the mouth or any really oral region. All of these viruses are capable of basically causing problems with cellular growth in these regions of our body. And that growth ends up being uncontrolled and leading to the development of tumors and cancers. The good news is we do have vaccines for HPV, the human papillomavirus, and that does help, or those vaccines do help protect against primarily strain 16 and 18 of these viruses, so that when people are exposed to them, they have less of a chance of developing cancer as a result. Papillomaviruses are very widespread. Almost every person on the planet will have been exposed to at least one strain of a papillomavirus at some point in their life, if not multiple strains. And again, most of the time, this is a situation where this virus is maybe inconvenient and causes some just kind of general irritation, but is not fatal. However, there are situations where certain strains of this virus can lead to fatalities as a result of the development of cancer. Our next virus is the influenza virus. There are actually a couple of different strains of this virus, or different types of the virus that can cause influenza. We are primarily going to be talking about what we call influenza A. There's influenza B and influenza C as well. But we are going to focus our studies on influenza A because that is usually the strain that causes the worst cases of the flu, as well as has the potential to turn into a pandemic where it spreads throughout the world and causes potentially millions of deaths in any given year. So influenza is a virus, and most of the time when people catch this, they have what we call the flu. Now, it's important to understand that the flu is a respiratory illness. Most of the time, symptoms include things like coughing, body aches, fever, congestion, and actually rarely do they ever include something like nausea and vomiting. Most of the time, when people have nausea and vomiting, they actually probably have something like norovirus or some other kind of foodborne illness or some other kind of virus that's infecting the gastrointestinal tract, not the respiratory tract. So technically there is no such thing as the stomach flu. That is a term that actually really means nothing. It's used a lot in society, but there isn't such a thing as the stomach flu. So it is important to keep in mind that influenza, the actual flu, is a respiratory illness that causes oftentimes severe coughing, congestion, like I said, body aches, fever, and potentially death. So I like to point this out for a couple of different reasons. One specifically is that when people are questioning the need for an influenza vaccine, for why they need to get their flu shot, oftentimes they misunderstand what the flu actually is. They're like, oh, I had, vomiting and diarrhea for a couple days and I was just fine. But most of the time that's not the flu. When we're talking about the actual flu, we're talking about a respiratory illness that has the potential to kill you. So, going on about influenza and some of the other important characteristics about it, this virus has a couple of important surface antigens. It has hemagglutinin and neuraminidase. Hemagglutinin has 18 subtypes, and neuraminidase has 11 subtypes. So when we look at this virus here, we have our hemagglutinin on the surface here, and then our neuraminidase here. Hemagglutinin is involved in causing primarily our red blood cells to kind of clump together. And neuraminidase is an enzyme that the virus uses for primarily entry and sometimes exit from the host cell as well. So both of these are important. The hemagglutinin causes just general damage to the body as it circulates throughout the body. And then neuraminidase is really what the virus uses to get into the cell and then to a certain extent get out of the cell as well, so it can go infect other cells. Now the reason I'm telling you this is because the combinations vary from year to year. So one year we might have a strain of influenza that is circulating that has H number 7 and N number 3. The next year, we might have H1N11. The year after that, we might have H3N7. So it varies from year to year. Some of these combinations are more infectious than others. Some of these combinations cause worse problems in the body than others. And this variation of the surface antigens from year to year is why we have to get influenza vaccines on a regular basis. It's also why some years the vaccine works very well and some years it does not, at least historically speaking. The way flu vaccines are created is that usually around sometime in the summer, various epidemiologists and microbiologists and virologists study the patterns of flu in various locations in the world. And they look to see what the most common strains are that year. And based on the evidence they have in the summertime, they work on creating the vaccine. So they make the vaccine based on what they predict will be the most common combinations in the coming flu season. It's not always possible to predict how a virus is going to mutate, however. In fact, it's oftentimes rarely possible to predict how it's going to mutate, especially when there are so many different possibilities of combinations. So some years, their predictions are very good. They are able to say with a good amount of certainty that this is a strain or these strains that we think will be prevalent in the winter months, and they create a vaccine based off that, and it works very well. Some years, their predictions are not as accurate, and that's not necessarily a failing on their part. It's just simply the way things work when you're dealing with variable viruses. So this is another situation where people say, well, I got my vaccine and I got the flu anyway, so the vaccines don't work. The reality is sometimes it's true, we miss the strain that does become most prevalent for various reasons, and so the vaccine was not as effective. Other years, it's great, it's really effective, but most of the time, even if you use a vaccine, you get vaccinated with a strain or a couple of strains that are not the ones that are most prominent in the flu season, you still have at least some protection, and even if you do get the flu after that, your illness is usually less severe. Now, in terms of treatment when people do get the flu, it really kind of depends on the strain and the person and what other kinds of things might be going on inside their body. Oftentimes, influenza is self-limiting. People will typically be sick for 2 weeks or so. They'll feel very bad for a week and then start to be maybe a little bit better after another week. So usually about 2 weeks, the worst of it is over. But the symptoms oftentimes last a full month when someone does actually get a case of influenza. So the first 2 weeks are typically really bad, and then the last two, their body's starting to heal and the signs and symptoms are starting to go away. For some people, though, they will require antiviral treatment and supportive care because the infection is starting to get out of control. And oftentimes, in addition to the viral infection, if it gets very, very bad, they will develop a secondary bacterial infection that will lead to additional lung damage and additional respiratory tract damage and will require antibiotics and additional care. In terms of prevention, there are vaccines like we just talked about a few minutes ago. Those vaccines vary in effectiveness because, as I told you, it's about 6 months before flu actually starts to circulate, at least in the United States in the winter season. That's when the prediction has to be made, at least as of right now. This is another one where I'm hoping with the advent of the mRNA vaccine, we can start to have a more rapid response with the flu vaccine. Usually it takes about 6 months to get enough doses of flu vaccine to go around, and that's why the prediction has to be made so early in the year. However, with mRNA vaccines that can be made much more quickly, we may be able to start creating flu vaccines later in the flu season so that they are more similar to the strains that actually will be going around when flu season hits. Another big one with this one is preventing droplet spread. So influenza is spread primarily through droplets when people do things like cough and sneeze. It can be spread on surfaces occasionally too, but most of the time we're talking about respiratory droplet transmission. And so it's really, really important that when people cough and sneeze, they cover, they make sure that they are not spreading those droplets to everyone else. The flu virus can oftentimes kind of linger in the air for sometimes up to 4 hours or so. And so it's possible that you could be walking through the halls at school. 4 hours ago, someone coughed or sneezed and left that in a droplet cloud for you. You breathe it in, and then you get sick and never even met the person who got you sick. Our next virus is Rhabdovirus. This causes a disease that we call rabies. Rabies is a very significant disease worldwide still. It is one that primarily infects non-human mammals, but definitely can spill over into humans and cause problems in a variety of ways. So in terms of transmission, rabies is mostly spread from mammal to mammal through bites. There are some exceptions to that. There are certain animals, especially cats, that may be able to transmit rabies through scratching, and some animals, like bats, can actually be so rabid, meaning they are carrying so much virus in their bodies, that even their saliva is enough to spread it to another animal. So most of the time, transmission is associated with bites. However, there are situations where it can be spread in other ways. In terms of the infection or the type of disease it causes, basically the virus is a very slow-moving virus. It takes a long time to migrate through the body, and it moves from the trauma site, which is basically the site of injection or the site of wherever it was that the virus made it into the body, the bite or the scratch, or sometimes even just being exposed to the saliva, like I said, of a bat, and getting into the body through some kind of mucous membrane that way. So it migrates from the trauma site to the nerve endings that are nearby, wherever that was. And then it very slowly makes its way through the spinal cord up to the brain. Once it makes its way up there, it starts causing a lot of swelling, a lot of pressure to build up, and does damage to the nerves in that region of the body. There are 4 distinct stages of rabies. The first stage is incubation. And in the incubation stage, what's happening is the virus is hanging out near the trauma site and starting to migrate to the nerve endings. In that process, it's also being replicated, so it's making copies of itself, and the viral load of the person, or the number of viruses in their body, is starting to increase. Eventually, when enough copies of the virus have been made and it has started to migrate up to the spinal cord in the brain, then the person or the animal enters what's called the prodromal phase. The prodromal phase typically has non-specific symptoms associated with it. What this means is the person is starting to get sick, but there's not enough information to know that what they're sick from is rabies. So they may have body aches, they may have a fever, they may just kind of in general not feel well, but there's nothing specific enough about those symptoms to indicate that they have rabies. Once they pass the prodromal stage, they enter into what's called the furious stage. The furious stage is the one that most people probably know the most about or associate with rabies. This is the stage where oftentimes the animal or even sometimes the person is agitated. They are starting to kind of froth at the mouth, and they are starting to show symptoms of what we would typically associate with rabies. So the frothing at the mouth comes from damage to the muscles in the area, in the facial region, basically, in the area that surrounds the jaw, and can also be associated with something we call hydrophobia. Hydrophobia means the fear of water, and rabies is sometimes called hydrophobia. If you hear an animal has hydrophobia, what they have is rabies most of the time. And the reason for that is the throat often becomes very sore and it's very difficult to swallow. And so in addition to starting to get dehydrated and creating bubbles of saliva near the mouth, the person or the animal doesn't want to ingest water and kind of wash all of that away and get some fluids in them because their throat is so sore and inflamed that swallowing becomes very painful. So saliva starts to build up. They're too afraid to drink water because of the fear of that pain, and so these bubbles start forming around the mouth. The next phase is the dumb stage. The dumb stage is basically when the person or the animal has had so much damage occur to their central nervous system, to their brain and to their spinal cord, that they start to lose cognitive abilities. They may still be a little bit agitated, but you start to see that oftentimes decrease in this stage, and basically their body's starting to shut down at that point. Their brain has become so damaged from this infection and from the inflammation and the viral replication that's been going on that the body can't do much more, and eventually most animals or humans are going to fall into a coma and then die shortly after that. Now, rabies is an interesting disease because it has such a long incubation and oftentimes prodromal period. So what this means is that, at least in the case of a person, and sometimes in the case of certain domesticated animals, if that person or that animal has potentially been exposed to rabies, there is still time to prevent the worst case scenario from occurring. What will typically happen, I'm going to use a person as an example here, is if a person gets bitten or scratched or potentially becomes exposed to rabies in some way or another, there will be kind of a two-pronged approach to treating the infection. First, they will receive an antibody treatment. So they are given pre-made antibodies that will give them passive immunity to this virus. Remember, we talked about that in Unit 3, where if you are given pre-made antibodies, you get protection from those antibodies. It may not last for a very long time, but it will give you at least some initial protection. So that antibody treatment is given to them usually in the form of an injection. Occasionally it could be an IV, but most of the time it's an injection. And those antibodies are there to help stop the initial spread of the disease. Around that same time, they are also given a vaccine for rabies. So they're given a vaccine that will allow them to make their own antibodies to that virus, so that if there is any remaining virus in their body after the antibody treatment has worn off, then they have their own antibodies that can fight off that virus. So these treatments, the antibody treatment and the vaccine, are designed to be used early on in the infection. They're designed primarily to be used in the incubation stage, but they could potentially be used in the prodromal stage as well. If someone enters into the furious stage of the disease, there is not a very good prognosis. Rabies has a 99.9% fatality rate in humans, and once that threshold from prodromal to furious is crossed, then most of the time people are not going to survive. However, in the past couple of decades or so, maybe I'll say 10, 15 years, some progress has been made. And they have found that in some cases, if someone progresses into the furious stage and you kind of catch it soon enough, you can put them into an induced coma, which essentially lets the body and the brain rest. During that time, you could potentially give them supportive care and other maybe antiviral medications as well to try to slow the replication of the virus. But the coma really just lets the body rest. It helps keep brain activity low. And in many cases, that seems to kind of let the virus run its course without the body overreacting to it. And eventually the virus will kind of clear itself out, or the body will help clear the virus out without doing too much damage in the process of getting rid of that virus. This doesn't always work to help someone survive this disease, but they have had some success with it. And there are a handful of people now who have survived a rabies infection that has made it to the furious stage by using this induced coma method to kind of let the body rest as the viral infection takes place and gets cleared out and the person can recover. In terms of rabies prevention, of course, there are vaccines that are given to domesticated animals, whether they're household domesticated animals or ones that we use in livestock. And it's a good idea to limit your exposure to animals whose vaccine status you are unsure of. People who work with animals on a regular basis tend to get rabies vaccines. This isn't a vaccine that's given to the general population because as long as the animals we come in contact with on a regular basis are vaccinated, there's no real need for that vaccine. However, people who do work with animals on a regular basis do tend to be vaccinated against rabies. We're going to spend the next couple of slides talking about the human immunodeficiency virus, or HIV. Now, this is the one virus that I do want you to know a little bit more about the structure than the others. One thing that's important to understand about this virus is that it is a special type of virus called a retrovirus. And this is significant because of the way it influences infection in humans. So, HIV is an RNA virus. That means that its genetic material is encoded in RNA. And when this virus gets into a human cell, what it ends up doing is using an enzyme called reverse transcriptase to take the information in RNA and make DNA. And that piece of information is significant in multiple ways. But one of the ways it's important is that, remember, that's the opposite direction that genetic information usually flows. So remember, usually genetic information usually goes from DNA to RNA to proteins. This is an exception to that, and it goes from RNA to DNA, and then that DNA is used to make more RNA and proteins. So it's a little bit different than usual, and there are some extra steps in terms of making viral proteins. Another reason this is significant is that once the viral RNA is used to make DNA, that viral DNA oftentimes gets incorporated into the chromosome of the host cell. What that means is that the viral DNA becomes a permanent part of the DNA of this cell or of that human. And that leads to what is essentially a permanent infection. When we have viruses that do things like cause colds or even certain papilloma viruses or other types of viruses, the virus gets into the body, makes a bunch of copies of itself, and eventually it's cleared out. That is not the case with HIV in most scenarios, because what ends up happening is the RNA gets turned into DNA, and that viral DNA becomes a part of the DNA of that human who is carrying that virus. And so that virus has basically become a part of that person and leads to a permanent infection. Now, most of the time when HIV infects the body, it infects white blood cells. And white blood cells, of course, are important in immune function. We talked a bit about B and T cells, which are the ones that are involved in doing things like processing antigens and making antibodies. But there are other types of white blood cells as well that are significant in terms of protecting the body from infection. So this virus ends up infecting cells that are critical to us being able to protect ourselves from other infections. What ends up happening is when enough white blood cells have been infected and enough virus has been created, then the body is susceptible to infections that it normally isn't susceptible to. So it is susceptible to fungal infections, bacterial infections, certain types of cancers, certain other types of viral infections, etc., that normally a human would be able to fight off pretty easily. But because their immune system has become so damaged, they can't fight off those types of infections any longer. So before we move on to just the last point on this slide, I want to go ahead and just summarize and kind of recap again, just to make sure everyone's clear, because I've found over the years that there are a lot of misconceptions around HIV, and a lot of people have been given a lot of incorrect or incomplete or sometimes even just bad information about the virus. And so this is an important one to spend some extra time with. So HIV is a special type of virus called a retrovirus. It is an RNA virus that uses a special enzyme called reverse transcriptase to make DNA. So that's different than normal, and that's significant because that influences the fact that this can turn into a permanent infection, and often does. So once the virus infects the body and the viral RNA makes it into the host cell, reverse transcriptase is used to copy the information in RNA and make DNA. That DNA that was made, that viral DNA that was created from that RNA, most of the time then makes it into the chromosome or into the genetic material of this host cell. So that means that this virus has become a permanent part of that person. It's become part of their own genetic code, at least in this cell, and the other probably nearby cells that this virus will go on to infect. What ends up happening after that is the viral DNA goes through the normal process of transcription and translation. Viral proteins are made, more virus particles are created, they are released from the cell, and they go on to infect other cells, and the cycle starts over. The whole process starts over again. And that means that the white blood cells in this person's body switch from performing their normal immune system functions to basically just becoming little viral factories. This ends up decreasing a person's ability to fight off infections that they normally would be able to handle with no problem at all. So in this case, it's not necessarily the virus that ends up killing them. It's the fact that their immunity is so low that there's some other kind of infection that comes and kills them as a result. Now, in terms of transmission for this virus, it is primarily transmitted through blood and blood products, but there are some other bodily fluids that can be associated. Things like semen and vaginal secretions can carry enough HIV to transmit that from person to person. And the virus can be transmitted from mother to fetus during the process of gestation, or from mother to infant during the process of breastfeeding. There is some evidence that the virus is carried in saliva, but at least as of right now, it seems to be carried in such a small amount that if just saliva is exchanged, there doesn't seem to be enough to cause infection in a susceptible person. However, if blood were also exchanged in that exchange, if blood were transmitted or associated with the saliva that was being exchanged, then that could be enough to transmit it to someone else. Now, the next thing to talk about that's important or significant when we are learning about this virus is that as the viral infection progresses, if it is untreated, eventually a person will develop what is called AIDS, or acquired immunodeficiency syndrome. AIDS occurs when an HIV infection basically meets certain criteria. Being HIV positive alone does not necessarily equal having AIDS. So there is a difference. Someone who is HIV positive may not have AIDS. However, someone who does have AIDS is HIV positive. So that's just an important distinction to make. And the reason for that is, especially from a medical perspective, is significant because at least these days, if a person receives proper medical treatment, receives the proper types of medications, it is possible to keep an HIV infection in check and under control so that they never develop AIDS. And if that is done, chances are very good that a person will be able to live a very normal life with a very normal lifespan. That was not true even just 20 years ago, but these days the medications are extremely good. They are very effective at keeping this virus under control in the body as long as they are taken as prescribed and taken regularly. And because of that, people with HIV, again, as long as they have access to these medications and can take them regularly, they can live very normal lives, healthy lives, and can live a relatively standard lifespan without developing AIDS. However, if someone has HIV and they do not have access to the medication or the medication doesn't work, the infection will eventually progress to the point where they meet the criteria to be diagnosed with AIDS. So again, just keep in mind that being HIV positive does not necessarily mean someone has AIDS. If someone has AIDS, they are HIV positive, but just because someone is HIV positive doesn't mean they have AIDS. And we can prevent that from happening. We can prevent someone who is HIV positive from developing AIDS with the proper treatment. So, getting into what actually counts as AIDS, there are certain defining characteristics of how someone is diagnosed with AIDS after they have become HIV positive. So, if someone is HIV positive and they have a Cd4 count of fewer than 200 cells per microliter of blood, they are considered to have AIDS. And CD4 is a certain type of white blood cell. It's a certain type of T cell that HIV is very likely to infect. Another defining characteristic of someone having progressed from just being HIV positive to having AIDS is having a CD4 count of fewer than 14% of lymphocytes total. So lymphocytes are certain types of white blood cells, and if we look at that group of white blood cells, the lymphocytes, and just look at the total number of CD4 cells that they have, if that's fewer than 14%, then they are considered to have progressed into AIDS. Another final defining characteristic of someone moving from just being HIV positive to having AIDS is having one or more of what we call the AIDS-defining illnesses. Now, there are several different AIDS-defining illnesses, and I don't list them in here because that list can vary depending on just what's going on in the world a little bit and whether there are new pathogens that are emerging or new information that we have. You can look up the AIDS-defining illnesses at any time on the CDC website, the World Health Organization, or any of the websites dedicated to providing information about HIV. But the AIDS-defining illnesses basically are conditions or diseases that people with intact immune systems don't tend to get because their body fights them off very easily. So certain types of AIDS-defining illnesses we'll actually cover in some of our future lectures, but certain types of fungal infections that most people usually do not get because their body's able to fight them off, or certain types of cancers, or certain types of helminthic infections that migrate to different parts of the body. Those are all considered AIDS-defining illnesses because it means that the HIV infection has progressed to the point that the person is so immunosuppressed that they are starting to get diseases that they normally would not because their body can't fight them off anymore. So again, this is just another recap of the information on this slide because, again, what I've noticed over the years is so many people have gotten incomplete, incorrect, or just straight out bad information about this virus. If someone is HIV positive, it means they are carrying the virus in the body. It does not necessarily mean they have AIDS. Sometimes being HIV positive does mean that someone also has AIDS, but only when they meet certain criteria. Any one of these three criteria means that an HIV infection has progressed into AIDS. However, simply being HIV positive alone is not enough to say that someone has AIDS. If someone does have AIDS, they are HIV positive. But again, one more time, being HIV positive alone does not necessarily mean someone has AIDS. And these days, if someone has access to medication, they will very likely be able to control their HIV infection to the point that it will never progress into AIDS. They can live what we would consider a relatively normal life. They can have children if they would like to, and they will live a pretty standard human lifespan. However, if a person does not have access to medication or chooses not to treat their infection, HIV will eventually develop into AIDS. And once someone does develop AIDS, they will die from that condition. Most of the time, they have 5 to 10 years at that point. That stage is getting a little bit longer these days with treatments that we have and knowing more about the disease and the virus and the types of infections it starts to lead to. But once someone progresses, at least at this point in time, once someone progresses into the AIDS portion of being HIV positive, they will end up dying as a result of this disease or of this infection. In terms of prevention and treatment, one of the big things to do in terms of preventing HIV is use condoms and prevent bodily fluid exchange during sexual interactions. Avoid sharing needles. So there have been many programs throughout the US and the world when possible or when feasible to do things like needle exchange programs so that for people who are going to use IV drugs, they're not sharing them with each other. There is a kind of newer medication, maybe within the last, 5 to 7, maybe even 10 years or so, called PrEP. That is a medication that people who are at high risk for developing HIV infections can take to help reduce the risk of getting HIV. So it's not something that people who are not at high risk for HIV infections would take, but for people who are engaging in activities that make them more likely to be exposed to HIV and more likely to contract HIV, there is a medication that can be taken to reduce the risk of developing that infection. In terms of treatment, we use a variety of what are called antiretroviral drugs. There are a couple of subcategories of these antiretroviral drugs. There are reverse transcriptase inhibitors, protease inhibitors, integrase inhibitors, and fusion or entry inhibitors. And each of these different subtypes works in a different way to help prevent the virus from replicating or getting into host cells or getting out of host cells. Reverse transcriptase inhibitors block the enzyme reverse transcriptase from working. So remember, when this virus infects a cell, it carries its genetic information in RNA, and then that RNA has to get transcribed basically into DNA, which is different than normal. An enzyme called reverse transcriptase is involved in that process. If we can block that enzyme from working, then we can stop the viral RNA from being turned into DNA, And if we can stop that DNA from being made, we can stop kind of the rest of this infection cycle from progressing. Protease inhibitors block capsid formation. So even if some of the viral DNA does get transcribed into DNA and makes it into the stage where, synthesis and assembly are going on, protease inhibitors will stop the capsid of the virus from being put together, which helps prevent fully formed viruses from being able to get out of the host cell and going to infect other host cells. Integrase inhibitors prevent viral DNA from entering host DNA. So they stop basically this stage right here. These are ones that prevent any viral DNA that has been made from being able to insert itself into the chromosome of that host cell. And that way that viral DNA does not become a permanent part of this person's genetic material. Fusion and entry inhibitors are ones that prevent adsorption. So these are ones that block the stage where the virus actually needs to attach to the host cell on the outside. This helps, at least ideally, would help prevent the virus from being able to even get inside the host cell and start the viral replication cycle at all. And most of the time when someone is being treated for HIV, they take at least 2 different types of medications. Sometimes they might take a reverse transcriptase inhibitor and a protease inhibitor. Sometimes they might take a protease inhibitor and a fusion inhibitor. They might take an integrase inhibitor and a reverse transcriptase inhibitor. Or they might take a couple of one type and one of another type. Basically, the combination can kind of vary. Their specific regimen can vary from person to person or just vary over the course of a person's lifetime, depending on what types of medications seem to be working at that point in time to keep the infection in check. It is important to understand that right now there really isn't a cure for HIV. Every once in a while you will see a headline about how HIV has been cured, but really the only couple of times that has happened legitimately is when someone has had an HIV infection and they've also had some kind of complete bone marrow transplant. So what they will have done is gotten all of their bone marrow wiped out and gotten new donor bone marrow stem cells injected. Those stem cells are not infected with HIV, and as a result, they develop a whole new set of white blood cells that are not infected with the virus. So keep in mind that is a very rare event. Right now, there is no real medication that cures HIV. That could change tomorrow, but as of right now, as of the time of this recording, there's no real cure for HIV. There are preventions for it, there are treatments for it, but there is no cure and no vaccine. Okay, we have made it to the point in the lecture we can start talking about COVID. I didn't start out the lecture with COVID just because of the type of virus it is. It fits into the enveloped RNA category, and although I've told you you don't have to memorize these and you don't have to, it just, again, helps keep everything organized and kind of flow along a logical route. So COVID-19 or SARS-CoV-2 or coronavirus, there's lots of different names for it, as you guys know by now. It is a member of what we call the coronavirus group. And the coronaviruses are a fairly large group of viruses. When you look at them under the microscope, it looks like they are wearing a crown, which is why they are called the coronaviruses. There are many different individual types of coronaviruses out there, some of which infect only animals, some of which infect only people, some of which seem to be able to be shared between people and animals. And these coronaviruses can cause everything from a general kind of mild cold to death, as we have seen. It is variable depending on the exact strain of coronavirus we're talking about, and also the individual who becomes infected. In terms of transmission, it is transmitted primarily through respiratory aerosols and droplets. Most of the time what ends up happening is someone coughs or sneezes. That is sprayed through the air. The virus ends up getting into the eyes, nose, or mouth of a recipient. That virus will then start to multiply in various cell types in the human body and cause a variety of symptoms. Again, as we have all seen over the last couple of years, that can be anything from a relatively minor cold to something more severe to ultimately death. So again, it is variable. It depends on the specific strain of coronavirus. It depends on the individual who has been infected and a variety of other factors. Right now, people who are getting COVID-19, many of them are able to treat themselves at home. It can be a self-limiting illness. They can take things like ibuprofen and other types of medications that will help ease the symptoms so they're not quite as uncomfortable, and then let the infection run its course. For people who have more severe infections, they may be treated with some type of medication. They may be given, one of the most common treatments right now is an antibody treatment, a monoclonal antibody treatment oftentimes. The way these work is, again, we've talked about these in Unit 3. This is a form of passive immunity where the antibodies to the coronavirus are made either in the body of another person. So let's say person A gets coronavirus, they survive it, and they are making antibodies. They can donate plasma, which will contain those antibodies. And those antibodies can basically be harvested and put into a medication form and given to person B who is currently suffering from coronavirus. That will give person B at least short-term protection to the viral infection. Hopefully that will help at least ease some of their symptoms, ease some of the damage that has been done in their body or that could be done in their body, and allow them to get through, hopefully at least, the worst of the disease until their body can start to recover. We can also make antibodies in a laboratory setting now that we know what the antibodies look like, and so we can develop them independently from having to acquire them from donations of survivors. That is one of the most effective treatments at this point in time. There have been other things that have been investigated that I'm sure you guys have all heard about, but at least right now, the best one that we have is some kind of antibody treatment. Although there are some medications that I think just in the last couple of weeks have been approved and released to help with infections as well. In addition to some kind of antibody treatment or potentially antiviral medication, typically people will also be provided some kind of supportive care, things to help reduce the symptoms and assist their body with maybe something like breathing when it is having difficulty doing that on its own. For prevention, the best thing is to limit exposure and get a vaccination. So what we have found after the vaccines were rolled out were that the majority of severe cases were in people who were unvaccinated, and that people who were vaccinated, even if they did get an infection, it was a relatively minor one for the most part that they could treat at home, and it acted more like a cold in that situation. So the vaccines have been found to be safe and effective, and they are saving a lot of people's lives and preventing a lot of illnesses at this point in time. Other information about coronavirus, I know there's been a lot of speculation as to whether this was truly something that was transmitted from animals to humans, or whether it was a lab accident, or whether it was an intentional bioweapon. And the reality is, right now, the best evidence that we have is that it was just a natural transmission from animals to human. Again, there's a lot of speculation about it being a potential bioweapon that was either one accidentally let loose from a lab or it intentionally released from a lab. But right now, there just isn't enough evidence to support anything like that. And we have seen coronaviruses make the jump from animals to people multiple times in the past. So with the first SARS outbreak and another one called MERS, MERS, these were other instances where the coronavirus jumped from an animal to people and started causing severe illness and death as well. So right now, that really does seem like the most likely scenario or the most likely case of events. There will always be speculation surrounding this virus, I imagine, but as of right now, the evidence suggests this is a natural event where a coronavirus jumped from an animal to a person the way it's done multiple times in human history. We have just one last virus to cover before we go into prions and then finish up this lecture. So this last virus we're going to talk about is one that causes the disease called polio. Technically, the disease is called poliomyelitis, but most people just abbreviate it as polio, and if you just said polio, everyone would know exactly what you're talking about. This virus is transmitted in a lot of different ways. It can be transmitted through food, through water, through direct contact, through fecal matter, and through vectors as well. And vectors are most of the time some kind of living animal that transmits it from person to person and doesn't really become infected by a virus or another type of microbe in the process. So different vectors, especially in this case, could be something like a housefly that transmits it from fecal matter to food. That's one of the most common situations when we're talking about vector transmission of polio. The way polio works is that it is ingested orally in some way or another. It starts to multiply in the GI tract. It is shed into the feces, and then it starts to move from fecal matter into the blood of the individual. So basically it leaks out of the GI tract, gets into the bloodstream, starts to travel through the bloodstream, and begins to infect nervous tissue. Now, for many people, this will be a relatively mild, self-limiting illness, which again means it will cause some mild symptoms that many people may not even recognize they've been infected with it, and the illness will go away on its own. The virus will basically run its course, the body will clear it from itself, and they will go back to normal. However, there are people that develop a much more severe case of polio called paralytic polio. So polio comes in kind of two varieties. There is paralytic polio versus non-paralytic. And non-paralytic is the version I was basically just talking about. This is the one that is usually a relatively mild, self-limiting illness that goes away. Some people may not even know they were infected with it. Paralytic polio, though, is much worse, much more severe. What ends up happening in this case is that essentially the person's body becomes so damaged by this virus that their muscles become paralyzed, and they will oftentimes go into cardiorespiratory failure without external support. So the nerves in their body become so damaged that the nerves cannot interact with the muscles properly, and therefore the muscles cannot go through the appropriate contraction and relaxation cycles. And this is particularly problematic when we're talking about the muscles that are involved in lung function and heart function as well. For these people, if they don't get proper care, especially soon enough, they will end up dying because they won't be able to breathe, their heart may not be able to beat properly, and their body will just simply shut down. One of the ways of treating this, at least for a long time, was by using what was called an iron lung. This was a machine that someone was inserted into and basically did the function of breathing for them by causing the contractions and relaxations, essentially forcing the lungs to contract and relax so that oxygen could get into the body as a result. In addition to potential things like muscle paralysis and cardiorespiratory failure, oftentimes people with polio, especially worst cases of polio, will develop muscle pain, spasms, inflammation of the meninges, and just in general have a really rough course of the disease that many people don't actually end up surviving if it gets to be this bad. Polio is preventable with a vaccine. There are two different types of vaccines that we can use. One is an oral vaccine and one is an injection. Both of them have worked incredibly well to get rid of polio in many places of the world. And at this point, most of us probably don't know very many people, if anyone at all, who has had polio. It's starting to get to the point that people who were alive before the vaccines were implemented and may have gotten polio are our grandparents or our great-grandparents. And so it's not something that is very common these days or something that is even talked about that often anymore. However, there are still multiple places in the world where polio is what we say endemic, meaning it is a disease that is found in those populations just on a regular basis, and people are contracting polio regularly in those communities. Lots of vaccination efforts have been done, but polio has not been able to be eradicated completely. With the decline in vaccination rates, we are starting to see more and more cases of this crop up, even in places we might not necessarily expect it to. And if we don't return to vaccinating a substantial portion of the population, we may end up seeing this as a disease that causes problems again in the future. To wrap up our lecture on viral and prion diseases, we are going to talk about the prions. I know we've talked about this a couple of times before, but remember, prions are infectious proteins. The diseases they cause are oftentimes called transmissible spongiform encephalopathies. And that is a fancy way of saying a condition that causes brain swelling and holes to form that can be transmitted or spread from person to person. These are considered neurodegenerative diseases because, as we've talked about in terms of the mechanism of disease, prions are altered version of normal proteins. So our normal proteins in our brain do things like hold cell membranes together, they hold microtubules together, etc. When we come in contact with a prion, what that prion does is get into our body and it starts to cause our normal versions of proteins to change their shape. As they change their shape, they start to lose their function. Things like protein tangles start to form, the microtubules start to fall apart, the cell membranes start to fall apart, and basically the cell that they have infected starts to die. If that happens to one neuron, that might not be a big deal. But the thing about prions is they don't just infect one neuron. They go on to infect the neuron that's next to the first one, and the neuron that's next to that one, etc. So even though these are just pieces of protein and they don't replicate on their own, what they do when they get inside the body is they basically force our normal proteins or our normal version of that protein to turn into the prion version of that protein. So if this is our healthy brain with stable proteins, normal neurons, and we get infected by a prion, that prion starts to cause our normal proteins in this neuron to change shape and essentially turn into a prion. And then all of those copies of normal proteins will go attach to the proteins on the cell next to them and cause those to turn into the prion version of the protein. And so this ends up spreading throughout the brain. While that is happening, our immune system is also notified that we have some abnormal activity going on. And so in addition to the prion itself basically making other prions form and causing damage that way, our immune system starts to try and clear away all of these protein tangles and damage that has occurred. And in the process of doing that, again, it does cause some damage as well. Just like with other types of infections that we've talked about before, oftentimes our immune system's response can cause more damage than the microbe itself. In this case, most of the damage is caused by the prion, but the immune system, in the process of trying to clear the damage that the prion has done, also does some damage. What ends up happening is the cells start to fall apart, and we start to get big holes that form in the brain, which is certainly a huge problem. There are some kind of generalized symptoms to most prions that infect humans. They typically cause things like confusion, fatigue. They may cause difficulty speaking and walking. They could cause hallucinations. They may cause problems with muscle stiffness or rigidness. They may cause very rapid onset dementia as well. So there are some different specific symptoms depending on exactly what type of prion we're talking about. But in general, when someone is infected with a prion, they will start to become confused, they will start to have fatigue, they will have difficulty speaking and walking, etc. Because literally their brain is starting to fall apart, and so they start losing all of those functions that the brain was controlling. Now, there are more prions than this, but I've picked out kind of the most common ones to talk about here. One thing that's, at least I think, kind of neat to know is that there are prions that infect all kinds of different organisms. So there are prions that infect people. There are prions that infect things like sheep. There are prions that infect elk and deer. There are prions that infect yeast. There may even be prions that infect bacteria. So it is kind of an intriguing type of microbe to look into and to study, because it appears that pretty much most, if not all, life forms on the planet have some kind of prion that does infect them and cause them to become damaged. We are just going to focus on some of the key ones for humans, though, and we'll start with one that's called Creutzfeldt-Jakob disease, or CJD. There are approximately 300 cases per year in the US, so this isn't a super common thing, which is good news because, at least as of right now, prions are always fatal. There's no way to not die from a prion. About 15% of those 300 cases are due to an inherited gene. So it's a situation where we can start to trace it through families. We can say, okay, parent A had this disease, and from their children or their offspring, some of their children, maybe 2 out of their 4 inherited this gene, and they will also eventually get Creutzfeldt-Jakob disease. So by being able to understand that at least in some cases this disease is hereditary, we can give that people information and they can make decisions based on that in terms of whether they want to have children, who they want to have children with, how they want to have children, etc. The worst news about this one, though, is that 85% of the cases seem to be sporadic, meaning we have no idea how people came in contact with that prion. We don't know where they got it from. We don't know why they have it, and someone that they lived with, maybe that shared pretty much everything of their life, doesn't have it. We just don't know. So prions are terrifying in some ways because in at least some cases, we have no idea where they came from. And when we don't know where they come from, we don't know how to prevent it. It's, at this point, what we can only say about it is it's bad luck. Hopefully in the future, we'll know more about how prions, or certain prions at least, can be transmitted, how we can come in contact with them, how we might be able to prevent them. But right now, it's still a bit of a mystery. Our next prion disease is called Kuru. It is actually the first prion disease that was discovered and was the disease that helped lead to the understanding that prions exist and they can cause damage. It is, as far as we know at least, transmitted exclusively through cannibalism in certain cultures or certain communities in areas of Southeast Asia. So this disease is transmitted when communities eat primarily the brains. It could be other organs as well, but primarily the brains of their deceased relatives. What ends up happening in some cases is someone will get Kuru, they will die, and then their family members will consume parts of their body as part of their mourning ritual or their grief ritual. And by doing that, what ends up happening is the family members who are exposed to especially the diseased brain will then ingest that protein, and the process will start over again. That protein will start to damage that person's body, and that person's brain will lead to another deceased person, and then their family will consume their body parts again through their grief ritual or their mourning ritual, and as a result, the cycle will continue. So a lot of communities have stopped this practice. It may happen occasionally now, these days still, but for the most part, it seems that once the communities figured out that they were transmitting a disease this way, they decided to enact some different rituals to grieve their dead relatives and show respect to them. Another kind of interesting fact about Kuru is that one of the symptoms is oftentimes uncontrollable laughing. It is a prion that damages the part of the brain that is related to laughing. And so one of the symptoms, it's not the only one, but one of the symptoms is that oftentimes people with KUR will start to just laugh uncontrollably at anything and everything or at nothing because just that part of their brain has been eaten away and destroyed by this protein. Our next one is called fatal familial insomnia, and this is the one that usually scares people the most because most of us experience insomnia at least a little bit at some point in our lives. And so once people find out about this one, every time they can't fall asleep, they start to worry that they might have this disease. So this one is, as far as we know, exclusively genetic. We don't have any situations where we've found a case that seems to be able to be passed from person to person. But basically what happens is a person will be carrying this gene, and most of the time, sometime in their 40s or 50s, the gene becomes active, starts creating the prion version of the protein, the normal protein, and then holes start to form in their brain. This particular one seems to work primarily on kind of the sleep center or regions of the brain that regulate sleep. And it starts out with not sleeping well and leads to a situation where a person can just never fall asleep again. Usually once it progresses to that point, the person has maybe 6 months or so to live. It's typically a very uncomfortable 6 months, and the best thing that medical science can do for them at this point is just try to make them as comfortable as possible, try to relieve the pain, try to relieve the issues that come along with not sleeping, and basically just wait for them to die, unfortunately. Now, like I said before, this disease or this condition seems to be exclusively a genetic situation. It's found in about 40 families worldwide, although more research has indicated it might be found in maybe up to 100 or so families at this point. And unless you are one of those families, unless you are part of that genetic lineage, you are not going to develop this disease. So if you have trouble falling asleep, chances are very good it is not fatal familial insomnia. However, if you are one of the members of these genetic lineages, there is a possibility that you will develop this disease at some point in your lifetime. And then our last prion that we're going to talk about is what we call bovine spongiform encephalopathy. Most commonly, it's called mad cow disease, and sometimes you'll see it referred to as variant CJD. This prion came to our attention several decades ago when it was found that people who were eating certain lots of beef were coming down with prion diseases that didn't match any of the others that we had known. And so before this, it was not well known whether prions could be transmitted from animal to person, but this situation really showed us that it is possible to obtain prions from animals, especially through food production and food consumption. And it's led to at least some changes in the beef industry in terms of what can be fed to cows, how things have to be regulated, how they have to be inspected, etc. So it's gotten a lot better in terms of regulating and reducing the chances that we will pass a prion from a cow onto a person. It's not impossible for it to happen still, it's just a very unlikely event. So it's something that still could happen, just not very common these days. And with that, that is the last topic of this lecture. I usually try to end lectures with some kind of good news or something positive, but there, at least at this point in time, really isn't a lot of positive information about prions. Right now, they are always fatal. We don't always know where they come from. We don't always know how to prevent them. And it is just something that we have to hope that researchers are able to figure out a lot about in the coming decades.